In previous class we have completed until here. Now let's see gibberellins. These are the types of gibberellins. So until now 130 gibberellins are identified. These are diterpenoid compounds. What is that it means? It has 4 isoprene units. So what it will be? So we write as C20H32, right? So C20H32 called as diterpenoid because it has 4 isoprene units, right? They are synthesized in young leaves, roots, developing seeds. The biosynthesis of gibberellin starts with geranyl diphosphate. So it is the precursor molecule of gibberellins. It is converted into ENT kaurin and again it is oxidized to Ga12. So it formed Ga12. Okay. This Ga12 is the central precursor molecule for so many gibberellins. So it is the main central molecule. Okay. Which enzymes are utilized in this process? There is copylyl diphosphate synthase, kaurin synthase, Ga20 oxidase, Ga3 oxidase. So these enzymes participate in the gibberellins pathway. Remember them. Okay. I told you earlier in previous class that these auxins, gibberellin, cytokinin, they are stored as inactive form. So first they have to be inactive, then they have to be stored in the plant, right? So this inactivation process done by Ga2 oxidase, that is gibberellic acid 2 oxidase. Okay, so it inactivates the gibberellin, then gibberellin is stored in the plant. So what did you understand until here? These are inactive forms. What are they? Ga conjugates glycosylated forms. They are stored in vacuoles and cytoplasm. Okay, remember this. So Ga conjugates glycosylated forms are inactive forms. They are stored in vacuoles as well as in cytoplasm. Breakdown, I already told you this. So this inactivation process done by Ga2 oxidase enzyme. So active gibberellic acid converted to inactive gibberellic acid. So when required to the plant, how this transport happens in the plant? Let us see them. These are transported through xylem and phloem. It is not a polar transport like auxins. What is polar transport? Polar transport happen in auxins. What is the place of auxin production? I told you in previous class. Auxins are produced at the top. So they produced at the top and transported to down. How they transport? They require energy. How this process happens? Because here is low pH, that means more acidic. Here is high pH, that means basic or neutral. Okay, so if there is 5.5 pH, here is 7.5 pH, then these auxins move from the top to bottom. Okay, so this movement or this transport called as polar transport. So it won't happen in gibberellins. It is working like a diffusion, right? But here energy is required. Also protons are required. What are the protons required in polar transport? They are, one is Auxin protein, another one is LOX protein. So it happens in the auxins, not in the gibberellins. So we call it as polar transport. Right, I am removing this because it is no longer related to gibberellins. Physiological effects. So what are the functions of gibberellins? It elongates the stems. It promotes the internode elongation. But cell division is not happening. Okay. In which phytohormones cell division happens? Please comment. Seed germination. Assume this is a seed, okay? It is an alluron layer. So alpha amylase lies in alluron layer. This alpha amylase reacts with the starch and produces sugars. So this energy that comes from the starch useful for the embryo to grow. So what happened here? Gibberellins promoted the alpha amylase that converted starch to energy. So this energy is useful for the seed growth, seed germination. Okay. There is also one more function of gibberellic acids, that is flowering induction. Gibberellic acid promotes bolting and flowering. You already know what is flowering. So I explain bolting. What is bolting? When plant gets stress, that may be from light or temperature, so plant moves energy from unwanted parts to seeds. Why to seeds? Seeds are helpful for next generation. From which parts energy is moving? Yes, the energy is moving from unwanted parts. That may be stem parts or leaf parts. So the energy moves to seeds. So these seeds helpful for next generation. So let's assume while mother in threat, so before she going to die, she shares her energy to the offspring. So now mother may die or live, but children will survive. So this process is like this. Okay. The next function that gibberellic acid does is fruit development. It stimulates parthenocarpy, that means seedless fruits. If there is more gibberellic acid, then there will be no seeds. Okay. The next function is mechanism of action. First I have to tell you about DELA proteins. These DELA proteins suppresses the plant growth by suppressing the growth genes. So here gibberellic acid binds with the GID1 receptor. So that forms gibberellic acid GID1 complex. This complex degrades the DELA proteins. So now DELA proteins are suppressed. So now DELA proteins are in offline mode. What happens now? Growth genes will be functional. Now plant growth continues. If you remove your distractions, you will be successful. We have completed gibberellins now. Let's go to cytokinins. Cytokinesis means cell division. So cytokines are helpful for cell division, not cell growth. Okay. Cell growth happen by auxins. Examples of cytokinins are zeatin, kinetin, benzyladenine. Okay. The biosynthesis of cytokinesis happens from the precursor molecule, that is isopentenyl ATP. The enzyme utilized here is isopentenyl transferase. It is synthesized at root apical meristem. One of the most common natural cytokinin is zeatin. So please remember it. Okay. Also remember one point. Cytokinins are adenine derivatives. Okay. Please remember these 2 points also. Cytokinin stored as cytokinin leukocytes, riboside forms. The storage location is in vacuoles. Cytokinins also stored as in inactive form. Okay. They are also same, right? Breakdown. Why to break down? Cytokinins are degraded by cytokine oxidase. So it cleaves the side chains of the molecule and it makes it inactive. Cytokinin transport happens in xylem and phloem. It is happening from root to shoot. Why? It is synthesized at root. So it has to move from root to shoot, right? Physiological effects of cytokinins. It promotes mitosis. So it is promoting cell division. It is also delays leaf senescence. So leaves survive for more days. Okay. It is also promote the growth of lateral buds. Here cytokinin working like completely opposite to auxin. The ratio between auxin and cytokinin decides which part to develop. That means shoot to develop or root to develop. So their concentration decides which part to develop. So remember this also. It is also called as anti-aging hormone. High cytokinin is responsible for development of shoots. High auxin is responsible for the growth of roots. Mechanism of action. Cytokinin signaling involves 2 independent systems. So cytokinins binds with the histidine kinase receptor, that is CRE1. So it leads to the phosphorylation cascade and also leads to activation of response regulators. The next hormone is ethylene. It is a gaseous hormone, so it cannot be stored. So it is a unique hormone, right? Its biosynthesis happen in young cycle. The amino acid, that is methionine, converted to S-adenosyl methionine, that is SAM. Then SAM is converted to ACC, that is 1-amino-cyclopropane, 1-carboxylic acid. And by the action of ACC oxidase, ACC converted into ethylene. Storage. There is no storage for ethylene because it is in gaseous form. Breakdown. Due to it is a gas, it can diffuse into the atmosphere and it get removed from the plant. It can also removed by oxidation process. Transport. Due to it's a gas, it to transport from air spaces. So diffusion happens as intercellular air spaces. The functions of ethylene are it to trigger cell wall degradation. It converts starch to sugars. It also performs chlorophyll breakdown. Okay. There is a triple response role in ethylene. That is, it inhibits the cell elongation. So it is supporting horizontal growth, not vertical growth. Okay. Leaf abscission. So it is responsible for fruit ripening, leaf abscission, and stress responses. Mechanism of action of ethylene. Without ethylene, there will be CTR1 kinase suppresses response will be happen. With ethylene, CTR1 inhibited, EIN2 activated. So next EN3 transcription factors are activated. Next the genes that related to ethylene will be expressed. Okay. So what is happening here? CTR1 inhibited. That's why EN2 activated. That is responsible for EIN3 activation. So that ethylene is activated here. The last one that I tell you is abscisic acid. It is also called as stress hormone. Biosynthesis. Abscisic acid is synthesized from carotenoid precursors that happen in the plastids. The key enzyme that involves here is 9-cis epoxy carotenoid deoxygenase. So it is responsible for biosynthesis of abscisic acid. It is mainly produced in leaves, roots, Seeds. Abscisic acid is stored in vacuoles as abscisic acid glucose ester. It is an inactivated form. When plant requires this hormone, it will be activated later. Breakdown. That means catabolism. ABA is degraded by ABA8 hydroxylase, produces phasic acid. It is also same, it is transported through xylem and phloem. But there is a small exception, that is, it is also transported in guard cells. Why it is in guard cells? Because it is responsible for stomatal closure when there is stress conditions. Physiological effects. The main function is stomatal closure. When there is a stress conditions, means there is a drought, so there is no water, so it closes stomatal openings. Let us see how it works. ABA accumulates during drought stress mechanism, promoting calcium signaling. Next, potassium efflux is happening. Guard cells losing turgidity. That means they are becoming straight. So while they becoming straight, stomata is closing, so there is no openings. Efflux means potassium is going out. Seed dormancy prevents premature germination, so undeveloped seeds won't develop due to abscisic acid. Stress tolerance, already we have told. Plant helps to survive in drought conditions, also in salinity. That means it protects from the salt, okay? Also from the cold conditions. Mechanism of actions. ABA is binding to PYR, PYL, RCAR proteins, so that this binding inhibits the PP2C phosphatase. So it is indirectly activates the SNRK2 kinases. So now stress responsive genes are expressed due to this action. Hormonal crosstalk. Which hormones combinedly works one function? Plant hormones rarely act alone, so they combine regularly. Examples: Auxin plus cytokinin controls organ formation. So both are helpful. Auxin helpful in cell elongation, cytokinin helpful in cell division. So both are helpful in growth. Okay. Abscisic acid and gibberellic acid works oppositely. So if there is more abscisic acid, there will be seed dormancy. If there is more gibberellic acid, then there will be seeds germinate quickly. So there will be always seed dormancy versus germination match. Ethylene plus auxin. The both hormones promote fruit ripening. Crosstalk callous plants to fine-tune development. By this action, the work will be done smoothly. Summary. Let us see everything, okay? Plant hormones regulate the growth, development and stress responses. Key process includes biosynthesis, storage, transport, signal perception, gene regulation, hormone breakdown. Understanding hormone signaling is crucial for agriculture, plant biotechnology, stress tolerance improvement. We have completed everything. If you really understand my classes, please subscribe. I really need your support. Thank you for watching.